All right, so Yossi took you through the steps of the data coming off the sequencer, going to your aligned BAM, and now we're going to go from the aligned BAM to short variants, SNPs and indels. So it's going to be that part of your best practices workflow. So we should kind of talk about what a variant represents. So it's a genetic change in a person relative to the reference genome, right? Like the big output of the Human Genome Project was to have a reference that represents a typical human being, although admittedly that human being looks very European, but, you know, c'est la vie. And so with respect to that larger reference genome, you can have variants that are either yours, that are part of your germline that you were born with, that are in every single one of your cells, or they can be somatic variants, which are variants that are sometimes associated with cancer, but are not necessarily deleterious. And those are variants that might be in some of your cells or not. But all of these variants are going to represent with respect to the reference genome, because it's an awful lot of data to give the entire reassembled genome for a single person. And so what we'd rather do is store that data as the way that it is changed with respect to some reference. It gives us a way to compress that data into a smaller format, and it gives us a way to compare individuals to each other. So the current standard at the Broad, I should say, is the HG19 reference, or we call it B37. There's slight differences in the mitochondrial chromosome and the naming scheme. But the latest release from the consortium that puts together the human references is HG38. So all of our genomes are going to be moving over there. There'll be some implications for leftover and so on and so forth. But the idea is that we keep improving the assembly. And for example, like some of the repetitive areas around the centromeres have been clarified so you can get better alignment and so on and so forth. And so periodically, every couple of years, we do release a new reference. And you want to make sure that all your resources and all your files are all with respect to the same reference, or your pipeline's going to be throwing a lot of erroneous variants, unfortunately. So outside of the realm of human variation, a lot of organisms do have assembled references. The Broad Vertebrate Biology group did a lot of them. So, you know, there's dog, there's horse, I think there's elephant, and there's a lot of different mammals, certainly, and some other reptiles and so on and so forth. Unfortunately, if your organism doesn't have a reference, using the GATK best practices, it's going to be pretty hard to work with. If you find yourself in that scenario, there are some workarounds, and there's sort of a non-human sequencing area on the forum for people that have these difficulties, but hopefully we're all doing things that have a well-defined reference, because that makes everything else easier. So here's a lovely diagram illustrating the different types of variants. We've been talking a lot about small variants, which are single nucleotide polymorphisms in the germline case, single nucleotide variants on the somatic case, because they may or may not actually have multiple forms or be polymorphic. It might just be one variation in one particular individual. But these are variants that are going to show up at a single position, like one base in the reference turns to another different base in the sample that you're sequencing. Indels, which is sort of a short name for insertions and deletions, are going to span multiple bases, either with respect to the reference or the reads that you're sequencing. And what we have illustrated in this diagram is a deletion, so that if this is the reference, there are a couple bases here that are not represented in our reads. Once you get into larger deletions, typically more than a couple hundred base pairs, then we call them copy number variations. And we've illustrated here a homozygous deletion, which in the germline case means that both copies of the chromosome have that particular part of the genome deleted, or a hemizygous deletion, in which case only one copy has a deletion. And you can also see copy number gains, which are typically more common in somatic sequencing, but can also happen in germline for typically smaller spans of the genome. Sometimes copy number variations get lumped in with structural variation, but structural variation also applies to a wider class of variants, such as translocations. So Yossi pointed out the example in the cigar for that particular read, that luckily the read and its mate did map to the same chromosome, but if you had a translocation, you would see a lot of reads where the mate was on a separate chromosome, because the fragment that you get from your sample actually aligns to two different chromosomes with respect to the reference. These are they're a lot more common in the somatic case than they are in the germline case, again. And you can also see reads in your BAM that may be non-human. Either they map to a very small portion of the human genome very poorly, or they don't map at all. And that's what our PathSeq tool that's going to be made public soon, or at least the documentation is going to be made public soon. PathSeq is meant to identify non-human sequencing in your BAMs. And again, that's just to show that all of these cases can occur both in germline data and in somatic data, but they're going to take on slightly different properties because the germline variants are going to affect all of your cells. So with 100% certainty, you can sample one of those cells that does have the variant, whereas the somatic variants will be localized to certain subpopulations of cells. So here we have a screenshot from IGV, which is the Integrative Genomics Viewer developed at the Broad, which is a beautiful way to visualize BAM data, specifically aligned BAM data, because we have the reference here along the bottom. There's a RefSeq track, which tells you that we are in an intron. You can tell because it has a skinny line, whereas the gene itself will have a thicker line like this. And then the little arrows show the reading frame direction, and then we're giving the gene name here, which is notch 1. So all of these gray bars represent different reads. You can see that they're actually, well, I don't think any of the reads are actually shown in their full length in this band, but you can click on them in IGV and find out what the read length is. They should pretty much all be the same. And at the top here, we see the depth of coverage, which is just a nice histogram summarizing how many reads you have. It should be in agreement with the count of reads vertically. Here we've highlighted in purple 2 reads that belong to the same pair. Yossi said that our target fragment length is about 400 bases, but we'll see some that are smaller than that. And so, for example, if you had a fragment that was only 250 bases and you're doing 100 base sequencing, you'll see, nope, not with that math. So if you were doing 150 base sequencing and you had a 200 base insert, then you would see some overlap in those reads, which is what we see in this figure here. And it's important to know that they do belong to the same fragment, that they are a mate pair, because again, as we pointed out earlier, we don't want to double count that evidence because while it is 2 reads that both support that SNP, it's really only 1 observation from the DNA. So also in this coverage at the top, you'll see this red and blue bar, which is showing you the allele fraction at that particular point for a blue C versus a red T. So all those T's are added up, which is a nice way to summarize the allele balance at that particular site. It's a pretty good allele balance, if you ask me. And so there are a lot of different attributes that you can look at for particular variants in IGV to see whether you think that they're high quality or not. Like if you see some variants that are causing some strange results later on in your analysis, you can open up the BAM, you can look at the reads, and you can say, do all these things make sense to me? And you can click through the reads and get all the cigar information and things like that too, which is pretty helpful. So once we do our variant calling, the output is going to be in a variant call format or VCF file. And so it's got a header with a lot of different information. It tells you the format of this particular, or the file version for this particular VCF. Usually it'll tell you which reference it was aligned to, because again, you want to make sure that all your resources and all your outputs are on the same reference. And it should also define all of the different annotations that are used in the VCF. For example, like looking at this record here, there's a DP and an AF, and you say, oh, I don't remember what AF stands for in this context, and it says, oh, okay, so this is allele frequency. If you were looking at somatic, it might be allele fraction or something else, which is the difference between frequency for population or fraction just for a single sample. So all the abbreviations used should be defined in this header. And any filters that get used, which aren't used in these particular records. But VCFs support multiple different samples. So for example, here we have 3 different samples from the 1,000 Genomes Project. And we have position by position records that encompass all 3 of those samples. We give the genotype for each of those samples, the genotype quality, which is in that Fred scale that Yossi explained, trying to describe the confidence that we have for the genotype call that we gave versus the next most likely genotype call, and the depth for this particular sample. So the depth in the info field should be across all of the samples in your VCF, and then in the per sample info in the format field, you'll get the depth for each particular sample. I'm a little shocked that we can call a GQ48 homozygous reference call with one read, but you know, a lot of these are toy examples. Anything else? And of course, there's this column here with ID, which is the identifier for that particular variant with respect to the dbSNP database, which is nice to know because for any given sample, a lot of the variation that you see should be common. And so then you should be able to see a lot of positions in the VCF that have dbSNP IDs assigned if you're using that resource. And we'll talk a little bit in the filtering about looking at things that have already been seen versus things that are novel in your call set and trying to get a good idea of what that balance should be. So we talked about how in GATK4 now we have a lot of different tools for calling variation in different contexts, germline small variants, somatic small variants, and somatic copy number variants, and we'll talk about the workflows for each of those different types. So our classic germline SNPs and indels are done in a lot of disease studies because it's very valuable to draw from a large cohort to investigate a disease, to see how common a variant is in a population, because looking at a single genome in isolation, which you might do for a clinical context, is still only useful if you know how rare those alleles are. For example, if someone has a rare disease and you're looking for a causative variant, all the variants that you're looking at are still only interesting if you know that they are rare with respect to the population, because if it's a rare disease, it should be rare in the variant that causes it should be rare in the population as well. For a different type of study, you might use family data. Again, like in a rare disease context, if you see a disease that presents in the offspring but not in the parents, you might want to sequence all of them to try and figure out which of those variants is most likely to be causal. So being able to use population data either from a family or from a broader group of unrelated individuals helps you estimate the rarity of your variants, helps you detect de novo if you're using related individuals, and helps you account for different ethnic backgrounds. Like, for example, if we sequenced a big cohort with one Finnish person, there might be a variant in that Finnish person that looked like it was really rare. But in the context of a group of Finnish people, that would have a higher allele frequency. And so it's important to get a ethnic background that's relatively consistent for your samples in order to get a good idea of how common things are in the population that that person belongs to. So there are two ways that you could do variant calling on multiple people. You could call them all separately and then effectively just staple the calls together. But in that case, you'll actually find that you have an underpowered analysis compared to our preferred way of what we call joint calling across all the samples. So joint calling is going to improve your variant discovery for low coverage sites. So for example, if we sequenced each of these samples individually, for sample 1, we see that there's an A to G SNP, which only has 2 reads supporting the G. Our middle samples are reference, and sample N also has an A to G SNP, but again, only with 2 Gs representing the variant. So if you sequence either one of those samples alone, if they're high quality bases, 2 is probably enough to call them, but it's going to be relatively low confidence. And if you get unlucky and they happen to be low quality bases, that variant might not get called. But when you look at the evidence from all of those samples together, now that you've seen 4 reads across all of your samples, that's a lot more evidence that makes you a lot more confident in that variant. It also shows that it's unlikely to be just a sequencing artifact, because 1 read could admittedly be a sequencing artifact, 2 reads in the same individual still, but once you've seen 4 across your whole cohort, you can dramatically improve your confidence. And what I consider to be a more important ramification of being able to do this joint calling is that you can get a better estimate of the allele frequency because you know what the coverage is for all of your samples. So for example, here we're showing two different samples at two different positions that are common SNPs seen in dbSNP with these RSIDs listed at the top. Sample A has coverage at those sites, but does not actually have a variant, so sample A is reference for those two SNPs. Sample B has a little bit of coverage for the first SNP and no coverage for the second SNP. And so if you called these two samples separately, for sample A, you'd have the two positions with home ref, or homozygous reference genotype calls. And you'd say, okay, that looks good. And if you called sample B by itself, you might have enough reads to output a homozygous reference call for the first SNP, but you wouldn't output homozygous reference calls at all, because in the context of just a single sample, we only output variants. And so if everyone is homozygous reference, we don't output anything. So you can't distinguish between the two cases where there is coverage, but there's no variant, or there's no coverage at all. And so this becomes more interesting when you have a third sample that actually does have the variant. And then when you joint call them all together, it'll show that one sample does have the variant, and then you can be able to determine who has coverage and who doesn't. So our multi-sample calling right now is in what we're going to call GVCF mode, or reference confidence mode, which we'll practice in the hands-on sections. And in this updated version of our best practices, each sample is run through haplotype caller by itself, and it produces an intermediate output that we can hold on to and then combine with other samples later on, which is very useful for calling large cohorts, especially if you're going to do them in batches. For example, you have a pilot study in order to get funding for a grant, and then your funding comes in, and you do one wave because only one of your collaborators has sent you samples, and then you have to wait for your second collaborator to send you more samples. You can keep computing data the whole time. But with the old version, where we put all the BAMs into haplotype caller at the same time, it takes a long time to run. It takes actually exponentially longer to run that way. But then once you get one more sample from that, like one patient that finally signed the consent form, then if you want to add that one sample, it's really hard in the old way. But in the new way, you just generate this new GVCF intermediate file for the new sample, combine it with all the samples that you've already processed, and output a new call set. So the GVCF version, where we output genotype likelihoods in this intermediate file, scales linearly with the number of samples, which is quite wonderful. And so you can always hold on to those intermediate files and combine them together with as many more samples as you'd like later on. So somatic SNPs and indels, or I guess you call them SNVs in the somatic case, are similar in a lot of ways because they use some same algorithms, but the workflow itself is quite different. So one of the challenges in somatic case is that it's very, very rare to get a pure sample of the tumor and a pure sample of the normal, because the way that most tissues grow, the tumor cells are very difficult to isolate from the normal cells. So, and you also don't know which of those cells are genuinely cancerous and which of them just contain mutations that will predispose them to cancer down the line. So for example, in this diagram, we've illustrated these purple and blue cells as decidedly malicious, pathogenic cancer cells. But then there's this orange population that has some variants with respect to the normal cell, and it's going to predispose them to becoming cancerous, but they're not yet, propagating without bounds. They're not yet metastasizing. They haven't yet taken on the properties that we would call a cell cancerous, but they're well on their way to doing that. And within the tissue itself, these populations are interspersed, and it's very difficult for a surgeon to go in with a needle and say, no, just the worst of the cancer cells. And so what you're going to have in your BAM file are reads coming from fragments of DNA coming from each of these subpopulations of cells. And so that's one issue. The other issue is that you only want the somatic mutations, right? So you're going to compare them against the background of a normal tissue sample so that all the germline mutations that are common to all the cells are going to be thrown out, because the germline mutations aren't causing cancer in your other cells, so we don't worry about them. But in a lot of cases, you'll see contamination of the normal tissue with cancer cells also, Because again, the surgeon's trying to go in and look at the healthiest tissue, which is hard to isolate potentially. Or you might take a blood sample to get your normal cells. But in some cases, like here, we see some of these particularly pathogenic blue cancer cells are already metastasizing into the blood vessels, and you're going to see cancer cells that are circulating in the blood. And so you're going to get contamination of somatic mutations in what you thought was your normal sample in that way. So this adds a lot of computational challenges to somatic variant calling that we didn't see in the germline context. In the germline context, we saw an awful lot of signal, which are, just the variants that are common to all the cells in your body, and a little bit of noise from technical sequencing or, maybe a handful of somatic variants that do sneak into your bloodstream when we do the sequencing. But in the somatic case, you're going to see all that technical noise that you would have seen just from sequencing in general, and on top of that, you see contamination of the normal sample and the heterogeneity of the tumor sample itself. And so then all of a sudden, your signal-to-noise ratio is much, much worse, and you're going to have less confidence in your calls. And so we're also going to look at the copy number variation workflow for the somatic case. So copy number is all about coverage. There are structural variation tools, and like the one that we hope to make public within the next 6 months or so, that will also look at the breakpoints. But for the copy number tools that we have available, we're only going to look at depth of coverage, which is a stronger signal just for calling variation anyway. So this kind of goes back to the diagram that we looked at in the earlier slides where we had the homozygous deletion, the hemizygous deletion, and we looked at the number of reads that would be associated with each of those. So you expect to see, if you have a normal section of the genome, the coverage will be pretty consistent. If you have a hemizygous deletion, you expect it to fall by about half. If you have a homozygous deletion, you don't expect to see any reads there. And that sounds like a really easy case, right? But we know that there are many attributes of sequencing that are going to introduce bias. Like we know that areas of the genome that are rich in GC content tend to be copied slower because the polymerase is slower, and so they're going to see a little bit less coverage for that reason. If you're working in an exome context, and all of a sudden you have the bias of doing the bait capture for each of the coding regions, and that's going to introduce bias in your coverage. And so what we need to do is collect the proportional coverage and be able to normalize that across all the positions. And we want to do this for a lot of samples in order to create what we call a panel of normals, or a PON, because we're going to use that panel of normals to remove noise in the somatic samples that we're sequencing to try and say, well, this is what a normal variation is. This is what the normal depth bias is in a sample that's not cancerous. And once we account for that, anything left that's deviating from that coverage profile will be a copy number variation. And then once we have those deviations, we can define boundaries for them to describe segments where we actually see different copy numbers. So again, these are very large events associated with somatic detection. You will see copy number events in germline, but they'll be much, much smaller. So we're showing this as the total copy ratio. So like, how does this compare to a normal sample? And the vast majority of the positions are 1, so there's no change. But here there's a 1.5 copy ratio for this whole chromosome, it looks like actually, which indicates that likely one of the chromosomes had a duplication. So you have 2 copies on one chromosome, 1 copy in the other chromosome compared to 2 copies in the normal, which is going to give you that 1.5 copy number ratio. And then here we have another big chromosome that's below 1, which is going to have some sort of deletion. These don't have to be pinned to like integer numbers or even halves, because we're in the somatic sense. We might have subpopulations. We might have multiple copies of the chromosomes. We might have contamination and so on and so forth. So this is a pretty accessible example, but you will see copy numbers at a lot of different integers. And in the hands-on session, we'll look at allelic copy number too, which helps us determine exactly how many copies we have on each chromosome, refining our copy number estimate, right? So compared to just how does this vary with respect to a normal? Like, for example, it's very different if you have one copy on each chromosome, or if you had one chromosome that was knocked out and the other chromosome was duplicated, it's going to have the same read depth, but it is not the same process biologically. And so the allelic copy number tool will help us distinguish between those two cases. All right, having talked about all of the different tools that we're going to use to analyze this data, so he's going to talk about workflow description language and how we put all these tools together into pipelines.